Yeah, hello. Let's make a start. Good morning, good afternoon, good evening to all of you, and welcome to the third In Silico Toxicology Conference. My name is Andreas Bender. I'm a professor in Cambridge for cheminformatics. We also do In Silico Toxicology, and I will guide you through the program today. Yeah, welcome. So what is the aim of this conference? The aim is to foster the community across all areas of the In Silico Toxicology research. But it's not only a research, it's also regulatory aspects, for example, new methods, new types of data, right? And the question is, how can we actually put that to good use in order to improve the approaches in our field? Okay, I'll clean that up afterwards. So what is the history of this meeting? So we had 100 people in Cambridge a couple of years ago. That was still in person before COVID. 2 years ago, we had 300 registrations. And this year it was about 600 or so. And there's now this Zoom channel here, as well as a live stream on YouTube. And I'm really glad that we reached people worldwide in really very different countries. So this is the conference on Zoom here. There's an additional live stream, as I said. And we also record most of the talks. Please see the event website next week. So that will be accessible then also in the future. Yeah, there's lots to do in computational safety or toxicology, right? And I moved to the field only relatively recently, so to speak, come from a chemical area before. There's many endpoints, right? But there's too little data, right? We don't understand mechanisms in many cases. The question is, how do we use machine learning methods in this area? Which types of omics readouts give us information, right? There's legal requirements as well, replacing animal testing and consumer goods first, then agrochem and so on, right? So there's really pressure to bring in new methods. And that's what makes it very relevant and also very interesting to work in that field. As for questions, please type questions in the chat window if you wish, also along the presentation. And that is mainly on Zoom, but I will monitor the live stream as well on YouTube. So you can type questions there as well, or raise your hand after the presentation in order to ask questions. And please mute yourself during the presentation. I will check that as well. If you use Twitter, feel free to tweet as well. So the Twitter handle is here. It's Tox 2022. Okay, we'll just check the mute everyone. Okay, and if you wish to present at the next meetings, please let me know. So the idea is to make that an annual or biannual event. Please let me know in this case. I would like to thank our supporters. So that's the BTS, the British Toxicology Society, LASA Limited, the RSC, especially the CSAC subsection, as well as CATS, the Cambridge Academy of Therapeutic Sciences. Yeah, and without further ado, let's start the sessions. The first sessions will be on Consortia. Our first speaker will be Mathieu Vinken from Belgium. I will stop sharing here, and then Matt, the stage is yours. Oh, you're muted, Matt? Yep.
Yeah, I was about to say you need to unmute me.
Okay, very good.
All right, thank you very much. Let's pull up my screen. All right, so I suppose that you all see my screen. So good morning, good afternoon, good evening, wherever you are. My name is indeed Mathieu Vinken. I'm affiliated with the University of Brussels in Belgium, and I'm currently coordinating a European project, which is called ONTOX. Of course, we like acronyms. It is an acronym. It is an acronym for a very long name, as you can see on this first slide, namely Ontology-Driven and Artificial Intelligence-Based Repeated Dose Toxicity Testing of Chemicals for Next Generation Risk Assessment. Well, the overall goal of ONTOX, which by the way started in May 21 for a 5-year period, is to develop an animal-free and especially human-relevant strategy for the prediction of chemical-induced toxicity. Of course, this is a very ambitious goal, I would say, so that's why we had to set priorities when we started off more than a year ago. So we decided to focus on systemic repeated dose toxicity for the very reason that this uses a lot of animals. We also focused on 3 organs that are very frequently targeted in systemic repeated dose toxicity, being the liver, the kidney, and the developing brain. And within those 3 target organs, we have identified 2 types of adversities that are frequently related with systemic repeated dose toxicity. being steatosis and cholestasis in the liver, tubular necrosis and crystallopathy in the kidney, and then neural tube closure and cognitive function defects in the developing brain. Now, all of the methods that we are developing in ONTOX should as such be chemical agnostic, so which means that theoretically they should be applicable to any kind of chemical, but of course we are well aware of the fact that we will have to test, and this is what we are doing, the applicability domain, and for practical reasons, we intend to prioritize drugs, cosmetics, biocides, and food ingredients, but of course it's not limited to this. Now, obviously, the driving principles of ONTOX is the 3 R's in addition to the principles of 21st century toxicity and next generation risk assessment. I will not elaborate on this because I assume that most of you are familiar with this. Well, ONTOX is a very pragmatic kind of project in the sense that we don't claim to be like the next project that will come up with another type of in vitro models. No, we try to make as much as we can use of what is already out there in terms of methodologies, in vitro models, in silico models, and especially with respect to data. And any remaining data gaps are filled by setting up targeted in vitro and in silico tests. Now here again, this is quite ambitious, and we do have an elaborate toolbox available to achieve that goal, if you will. So the first aspect of that toolbox is human data and biological material. Secondly, in vitro methods, whether or not combined with stem cell technology, and then very important silico methods, and in particular, artificial intelligence. Now, the main deliverable, if you will, of the ONTOX project is to come up with these 6 NAMs, new approach or non-animal methodologies for each of the 6 case studies, as already explained before. Now, you might know that there are many definitions out there for a NAM. It's very important to be on the same page, and that's why I want to make the definition of a NAM in the context of ONTOX very clear now. So as you can see on this slide, so it is an artificial intelligence-based strategy linked with a battery of in vitro assays and in silico tools for hazard prediction, obviously to be combined with customized exposure assessment for the sole purpose of a human risk assessment. A very long definition, maybe better understandable maybe by looking at this slide. So we have 6 of these NAMs, and as mentioned, they consist of 2 components. On the left-hand side, you have the artificial intelligence system. I will be coming back to this further on. And then the second component on the right-hand side is the cell culture system linked with in vitro assays as well as with in silico tools. And also will be explaining this further on. Well, I would say that the main technological or methodological cornerstone of ONTOX is artificial intelligence. Again, I don't think I have to explain to you what artificial intelligence is all about. But basically, we are applying artificial intelligence in ONTOX for 2 purposes. The first one being the collection of big data. And we are particularly interested in data that relate to biology, toxicology, physical chemistry, and kinetics. Why? Because we know that these data are very important for systemic repeated dose toxicity. And we collect those data in a structure that we call the ontology. And I will be coming back to this further on as well. So there is no limitation in terms of the sources that we consult to collect those data. Obviously, the first thing that we do is to dig into scientific literature, but very important is that we do this in a transparent way, so hence the implementation of the FAIR principles. And then the second, and I would say the most challenging application of artificial intelligence in ONTOX is to use it for hazard identification or prediction. So what we want to do there is to create a kind of a signature, if you will, of the 6 toxicological effects that we have identified and that we actually prioritize. We do this by means of machine learning, more specifically a technique called read across structure activity relationship. And I will be coming back to this further on as well. So as mentioned already, ONTOX is pragmatic in the sense that we try to rely as much as possible on data that are already out there. And to do this in a kind of a structured way, we use 3 tools, as you can see here on this slide. So the first tool on the left-hand side are the physiological maps, and I will be showing you an example, which is fully fed or fully being based on mechanisms. The second tool are QAOP networks, so quantitative adverse outcome pathway networks that are not only being fed by mechanisms, but that also rely on kinetic data and also toxicological data. And this then serves as a conceptual basis for the third data collection tool, which you can see on the right-hand side, the ontology, which as such is the same as a QAOP network, with that difference that it also takes into consideration the physical chemical properties of the chemicals that induce that adversity. Just to give you an idea, so as mentioned, we started off more than a year ago. This is one of the first physiological maps that we have created thus far. What you see here, and I will not go into the details, of course, this represents vitamin D processing and metabolism in the kidney, which is one, of the physiological studies, or the physiological effects at least, that underlies one of the 6 case studies. This is then an example of a QAOP network. This is the QAOP network, if you will, for cholestasis, so which refers to the accumulation of bile acids in the liver. Again, I don't have time to go into the details. I assume that most of you are familiar with the context or at least the idea of an AOP. So on the left-hand side, you have the triggers of the toxicity, the molecular initiating event. And this then runs through a number of key events to eventually result on the right-hand side in the actual toxicological endpoint. And then very important, of course, is the ontology. And as mentioned, this is as such the same as a QAOP network, with that difference that it also takes into consideration the physical chemical properties of the chemicals that induce the AOP. And of course, as is the case for an QAOP in general, that it also gives you an idea on kinetic and quantitative relationships between the key events. Now, the second cornerstone, if you will, of this ONTOX project is the in vitro and in silico test battery. Again, I don't have to explain you what it's all about. Now, these are being generated as we speak. So we set up one in vitro and in silico test battery for each of the 6 case studies. And this is where we differ also from other European projects that have tried to do this in the past. So we go back to the very basics. So we go really, into the physiological functions that are of relevance for the adversities. So we dig into textbooks and so on. So this is where we get our information. And this then serves as the basis for setting up these physiological maps, as you have seen. So the second step is then, well, now you know the homeostatic, the physiological situation, but what if things go wrong? So what in case of toxicity, and this is where the QAOPs comes into play. I also showed you an example of this. And this will then allow you to identify the so-called key events. So the processes that are critical for that AOP to take place. The next step is then to identify and to select at least one existing in vitro or in silico assay to monitor each of the particular key events. And then, as you can appreciate from the final bullet point, what we need to do there is to find the ultimate culmination of all of these assays that give you a reliable prediction of that particular AOP. And as mentioned, this is what we are doing. This is work in progress. This is just an example of such an in vitro and in silico test battery that we are constructing. So it's again, it revolves around this QAOP network on cholestasis that I showed you previously. So as you can see, it basically includes a broad variety of in vitro and in silico assays. So for example, for the in vitro assays that really are located at different levels of expression. So the real expression level, but also the functionality level. And of course, the main challenge is to find the most optimal combination of all of these assays that will give you the most reliable prediction of that kind of toxicity. So this gives you an idea on the workflow that we follow in ONTOX, consists of 3 steps, 3 gears, if you will. As explained on the left-hand side, this is the stage where we are still in, is tier 1 data collection for setting up the ontologies. So this brings us to the second tier. And the ontology, as already explained before, not only helps to feed the artificial intelligence system, but as you can see from the slide, it also serves as a conceptual basis for setting up the in vitro and in silico test batteries. And of course, this will then enable us to do hazard prediction, and then together in step 3 or tier 3, if you will, with exposure assessment, will allow to do full-blown what we call probabilistic risk assessment. I will be coming back to this further on. Very important also is that from day onwards, we are already in contact with the end users of all of this. So that would be people in regulatory settings, but also in industry. Well, we distinguish 2 main stages, phases, if you will, in ONTOX. So the first, where we are still in, is the stage where we optimize all of the systems. So the AI system, but also the in vitro and in silico test battery. And obviously you want to do this with data-rich chemicals. So chemicals of which you know very well that they induce one of the 6 case studies and for which there is a lot of data available. Now, the ultimate challenge, of course, lies in phase 2, the so-called ab initio case studies. And this is where you want to test chemicals that are data poor, of which you only have a couple of clues that they might induce that particular type of toxicity, or no data at all. So how to deal with that? So the first thing you want to do is to apply the artificial intelligence system that you set up. You will use it as a kind of a benchmark. So you will compare what comes out of the artificial intelligence system with what we already have. And this will give you a probability of that chemical to induce that kind of toxicity. Now, in case of low probability, well, there's no need to follow up. So you can de-risk, so to say. In case of mid or high risk, then you need to follow up by means of this in vitro and in silico test battery that was previously explained. At the same time, in step 3, you also need to do customized exposure assessment. And this, as already explained before, will enable you to do risk assessment. And this is also part of the novelty of ONTOX. We step away from the classical binary risk assessment, so yes or no, in terms of a compound being toxic, towards probabilistic risk assessment. That really includes already an element of uncertainty. Now, it's a quite big European project, and as always with these kind of initiatives, consists of a number of work packages. There are 14 of them, as you can see on this slide. All of the practical, the research work, as explained, is being done in these, red and blue work packages, if you will, the first 9 work packages. And the idea is that these 6 case studies, which are being addressed in work packages 7, 8, and 9, so the red ones, run in parallel, first of all, through the different aspects of the ontology. So this is where we collect all of the data. Then in work package 5, everything is being integrated. And this is also where the artificial intelligence magic, so to say, happens. And then very important is that is being applied and implemented in work package 6. And of course, this is being framed, as you can see, by these green work packages that address or that, are devoted to other tasks, but nevertheless also very important, like coordination, data management, and so on. Just to give you some background information, practical information, we are 19, originating from 9 different countries, as you can see from the right-hand side. So not only European countries, but also the US. We are a growing community, so more than 100 researchers. Most of us have an academic background, but we also have people from industry. We are very fortunate, of course, to have a substantial budget available. And we are also very fortunate to be supported by a scientific advisory board with people, with different backgrounds. And this is exactly what we need, of course, because this is really an intersectoral kind of endeavor. Now, we don't do this job alone, of course. We actually team up with 2 other projects that started around the same time and that have the same goals, so to say. So that is Risk Hunter, which is coordinated by Bob van der Water, which, as you might know, is the follow-up of the UTOX risk project that ended last year. And then the second project, and I saw in the program that there will be a talk on this as well, is PrecisionTox, which is coordinated by John Colburn from Birmingham University. So we gather in this cluster that is called ASPIS. And again, we like to use acronyms. So by the way, ASPIS is the Greek word that stands for shield, so to protect us against chemicals. Again, it's an abbreviation for a very long definition, Animal-Free Safety Assessment of Chemicals Project Cluster for Implementation of Novel Strategies. It's a very huge, it's a huge cluster actually. So 70 partners from 17 different countries. And here again, of course, in view of increasing our impact and visibility, we are supported by different entities. So for instance, as you can see, we have this ASPIS Regulatory Forum, which is being led by the Joint Research Center. So to be, in contact with the regulators. We also are in contact and collaborate with other initiatives like PARC in Europe, as you might know, but also things, at the other side of the ocean. And here again, we also have a scientific regulatory advisory board that is populated by people with different backgrounds, not only Europeans, but also from the US. Well, this was ONTOX in a nutshell. I hope that this was interesting to you, and I hope that you want to follow up on ONTOX. So in that case, I invite you to consult our website, but in particular to subscribe for a newsletter so that you will be, automatically informed about the newest developments in ONTOX. And we're also pretty active on the social media, as you can see here. So with this, I would like to thank the organizers for giving me the opportunity to introduce the project here. And I would like to thank the audience for your attention. Thank you very much.
Great, thanks a lot, Matt. Much appreciated. And there's one question already. Nadav, do you have a microphone? Then we can make that a bit interactive. Then you can ask that orally and follow up on that.
I do have a microphone. Can you hear me?
Yeah.
Yes.
So my question is you mentioned in vitro assays and deciding which one you would like to do. Are these chemicals or AOP specific? I mean, do you choose them case by case, or are you planning to choose assays that will be applied to all cases for consistency?
I don't fully get your question. Are you referring to the chemicals or to the assays?
Will you be applying the same assays when considering different types of chemicals and AOPs that you want to study?
Yeah, of course. Well, there are generic key events in the AOPs, and then we are talking about things like inflammation, oxidative stress, which can be found in any kind of AOP. And obviously, for those, you will use the same assays throughout the different AOPs and for the different chemicals. But there are also quite specific AOP-specific key events, like, for example, in the case of cholestasis, you have the inhibition of the bile salt export pump and drug transporters. These are AOP-specific key events and also assays that you cannot use for other AOPs.
All right, thank you very much.
Yeah, and I guess in some sense, that's quite an important question, right? If you generalize that, we had that with gene expression data before as well. Do you do something that's consistent so you can generate models based on consistent data? Think about cell line, dose, time point. Yeah, there's always those three variables where you don't really know what to do in many cases, right? So do you do that consistently so you can build models, or do you do that in a specific way for the organ of interest? So you change the cell line, right? You know it's a different readout, so we need 6 hours instead of 12 or 24. Yeah, but we need a different dose that's maybe therapeutically relevant, right? But that makes your data inconsistent. Yeah, and that is, I found that also very difficult to deal with. Yeah, to make a decision. Yeah, consistency versus relevance. Yeah, there's a trade-off in many cases, right?
Yeah, well, it's a combination actually. So in the sense that the overall strategy is identical throughout the different AOPs and case studies, but of course the selection of the specific parameters that you just mentioned, like the dose, well, the concentration, I should say, the exposure regime, that might differ. But here again, we try to be consistent. Actually, we had quite some discussions on this. What would be the appropriate, concentration range for drugs? Should we consider Cmax? Should we go for the typical IC10? These kind of things. So it's a kind of a combination between, and this is part of the challenge, between trying to figure out what can be consistently used and what would be case specific.
That's right. Thank you. Are there any other questions from anyone?
I've got a question. It's Carolyn here. You mentioned in your phase 2, you wouldn't follow up on negative predicted compounds. Could you explain that?
Did I mention that we do not follow up? You mean if the, in case of the probability from the artificial intelligence exercise is low, that we do not follow up? Yeah, that is true. But that needs to be distinguished from the use of negative control chemicals. Because obviously, in everything that we implement now in ONTOX, we do pay a lot of attention to, negative controls. And negative controls can have different meanings. We are not talking about solvent controls, but for example, chemicals that induce another type of toxicity, but not the one that you're interested in. This is exactly what we use to build up our artificial intelligence system and also to test the robustness of the in vitro and in silico test battery. And that is why we would be pretty confident in the ab initio kind of stage. If something would turn out to be negative from the artificial intelligence system, which can be considered at the first year, that there would be no need to follow up by means of in vitro and in silico testing.
Yeah, I'm just wondering if it would be wise to initially confirm that. And I assume if you've got a lot of confidence in your model, but wouldn't you want to build that confidence first?
Exactly. This is why I explained that we have 2 stages in ONTOX. So the first one is where we actually, want to confirm this and actually build this model, and we'll test elaborately, I would say. And then in the final stage, and that would be probably the last year of the project, then we will challenge the system.
Okay, so it would be a later stage where you
obviously, yeah. And this is, by the way, also the reason why we tend to move to probabilistic risk assessment and not the binary risk assessment, because we fully consider this element of uncertainty.
Great, thanks a lot. Any other questions? We still have 2 or 3 minutes in case there's any other questions from anyone.
By the way, if you were to be interested, I'm of course more than happy to share this presentation. Just drop me an email, or this could go via Andreas.
Yeah, please just send it to me. I can put it on the website as well. Then it's in a central place. Yeah. Hey, David, you have a question?
Yeah, thank you, Mathieu, for the very comprehensive introduction. Here, I want to understand a bit better about this AI part. So in my understanding, is machine learning model that gives a probabilistic assessment of the likelihood of causing toxicity. And there, my question is actually twofold. One is how robust this will be, and how would you assess the robustness? And the other is actually how to prevent the common caveat that we meet in our daily work is that we are looking just under the light where we can find our lost keys, essentially the coverage. Thank you.
Yeah, well, of course, I'm not a specialist here in the artificial intelligence aspect of that. I'm a toxicologist, but we do have Thomas Hartung, who is leading that work package in ONTOX. What I can tell you is that the basic principle of the artificial intelligence concept, if you will, that we use is RASOR. So read across structure activity relationship, and you might have a look. There there are a couple of very nice papers out there. So it really consider like 100,000 of chemicals. Initially, it was said to only look at structure-activity relationship, as the name suggests. But as part of the work in ONTOX, we will also include, biological effects. And of course, given the fact that we have a look at so many different compounds, we are talking about 100,000 of compounds. I mean, this is a huge check of robustness. So that is, I would say, the very important step. And here again, to come back on the previous comments, this is also, of course, where we fully consider the negative controls and, the compounds that actually, don't give or don't induce the kind of toxicity that you're looking at. So that is also part of the robustness exercise. And maybe also of interest is we consider both structured and unstructured data, by the way, for building the RASOR approach.
Okay, excellent. Great. Thanks a lot, Matt, for your presentation and answering the questions. Much appreciated.
You're very welcome.
We will move on to our next speaker then. Our next speaker has a decade or probably even more of experience in building consortia in order to share data, build in silico models for toxicity prediction. That is Barry Hardy. Barry, feel free to take the stage.
Good morning, Andreas. Good morning, everyone. Can you hear me okay? Andreas, are you able to hear me okay?
Oh, yes, everything.
Oh, okay. All right. So in this presentation, I attempt to give an overview of the work that's been going on for over a decade with regards to developing open science knowledge infrastructure, and particularly as it applies to predictive toxicology and risk assessment. So I thought I'd start with a definition. There are a number of definitions from open science. This one is from the OECD a few years ago, emphasizing the importance of open science, being able to access scientific knowledge and data from public research. And you notice they emphasize the importance of collaboration, which is of course, as we just saw in Mathieu's project, a lot of our projects involve a lot of collaborative research. So the knowledge resource development, importantly, needs to support collaboration, and it also needs to sustain access to the data, but also other kinds of important aspects in knowledge development, including increasingly models, like we again just heard from the AI machine learning work in ONTOX as an example. So we also need to document models and protocols and sustain those too. And this portrayal here, I think, captures some of the complexity of this, because there are many different practices that across the life cycle, from when we first plan experiments, then how do we organize the documentation of the experiments, collecting data, processing it, and then making it available over the longer term, for example, to perhaps a different community So biologists might be generating the data. How do they document and organize that data so downstream it can be used by other users, such as risk assessors? So in summary, I think in open science, we have a lot of different practices to address. Of course, data management is a very high profile, important one. The data is probably the most valuable output from a lot of research activities. And if we are building models, we're building models on data. And I think probably all of you here have heard about the FAIR principles, where we try to improve our practices to make data findable, accessible, interoperable, and reusable. So these FAIR principles are important. And of course, ontology for this goal of organizing knowledge is important. And this little diagram shows even within open science, we have a language to describe. And it also covers various aspects of practices, including data, but also, as you see here, including documenting science in workflows, in notebooks, and tools, and so forth. And open science is growing very significantly. So we're not talking about one or two organizations, a few projects. We're talking about many organizations and many projects. So we want to sustain and integrate knowledge from many different stakeholders and many different projects. Now, if you go back to ONTOX, which was a project we ran from 2008 to 2011, there was very little in place at that time for thinking out a framework which would support goals like interoperability, components that could share a common language and talk to each other, and so forth. So this was our first attempt over a decade ago to lay out how we thought the thinking should be to have an interoperable, harmonized knowledge infrastructure. The first two use cases we ran actually live in Berlin around that time. Demonstrated making a prediction, a toxicology prediction, and building a toxicology model. And we ran the software across several different locations in Europe. So it was our first demonstration over a decade ago of this kind of concept. And I think this concept has proven to have a lot of good aspects to it that's been further developed on other projects. And today, in terms of operability, we have quite a lot of interfaces like this and tools to support databases talking to each other and talking to algorithms and so forth. We also at that time had some good discussions and a couple of papers, a perspective and a roadmap on the importance of ontology and a common language. And I think that topic is still very important today. And without this kind of common metadata, it's very difficult to do integrated data analysis. And so again, this I think continues to be a really important aspect. With regards to sustaining ONTOX, we sustained it as a nonprofit, international membership-based organization, which continues today. We've been focusing on educational activities with the COVID situation, running virtual conferences, virtual schools. We started an internship program. So ONTOX sustains this kind of community interested in open science in the area of toxicology, and now we include it more into the actual infrastructure development and deployment, as I'll talk towards the end of this talk a bit about what we're doing there. A more recent infrastructure project was OpenRiskNet, and there you'll see sort of some of the development from ONTOX. It's more driven by the services are driven by satisfying different use cases, and we also have the concept there that we need to try to interact between different use cases. So whether it's from starting with cheminformatics and data curation through to predictive metabolism, systems biology, developing quantitative AOP models using toxicogenomics data. So you can see that we've moved over this last decade from working on thinking out how the services should be defined and organized to implementing them more in terms of use cases like this. The EU Toxicology project, which ran 2015 to 2021, there we implemented quite a lot of activity, which is either open or becoming open. So including documenting methods related to in vitro experiments, especially. A lot of this data is now available through the EBI's bio studies. There's about 1,200 data sets. But you see here, we also worked on how to process that data, make it available through notebooks. You know, that effort continues today. When you have that data prepared well and available in a fair kind of way, it can now be used on some of the newer projects, including yours. We worked within EU Tox Risk on an important topic, working with data generators on how they should document their protocols and their associated data. And we had a first version of the data template developed there, which describes the compound information in the study. And we've been working on a second version, which we plan to release this autumn, trying to harmonize this more and structure the metadata, improve the metadata more, extend it to different kinds of experiments. So this is an ongoing effort. And I think also, if you're interested in this topic, one that really should be an open science one. So we want to improve it across different projects. Moving on, Risk Hunter it started last year and has the goal of developing solutions in next generation risk assessment, including the goal for use in industry and regulatory acceptance. So again, it becomes very important that everything is well documented, it's harmonized, the data is prepared well. And in Risk Hunter, you start to see now more increased use of modeling, toxicokinetics, exposure modeling, predictive metabolism. So we also have to think out, how do we document in silico protocols? How do we organize our descriptions of models for quantitative AOP goals? So there's plenty of new kinds of, let's say, knowledge infrastructure development work to do there. And Risk Hunter is within the context of this ASPIS cluster that Mathieu mentioned in the first talk. And there, we've had some really good discussions in the first year of the projects on how we could collaborate on resource development, supporting chemical selection, sharing databases like the chemical database and the methods database. And I think we've got a very good momentum there for collaborating together and setting up the processes for how we not just develop resources, but share them and deploy them and have the other projects use them. And I think a lot of discussion of our progress will be at this meeting, this open symposium in November. Just on the nano side, we took a lot of these ideas. I'll just spend a couple of minutes on how they've developed within the context of the nano safety community. So the different projects and organizations looking at how do we evaluate the risk and safer by design with regards to some of the more complex materials coming from nanotechnology and even material science. And so there we took these ideas and developed a workflow to integrate terminology from different existing ontologies so that we could have an ontology that covered the use cases needed in nano safety. And we're taking a similar kind of approach with that data template work I mentioned under EU Tox Risk. And then a follow-up project to eNanoMapper. eNanoMapper was 2014 to 2017. I think made nice progress on that knowledge infrastructure. and has been built upon within a more recent project that just completed called NanoCommons. And NanoCommons is a knowledge community infrastructure project. So the idea within NanoCommons was to bring different resources together, databases, modeling tools, QSAR modeling, risk assessment workflows, and bring it into a common infrastructure, knowledge infrastructure. One example is in characterization, which is very important with these kinds of materials. How do we go about documenting the protocols for characterization? So we've built, I think, a quite good resource that'll be part of the open science infrastructure goals I'll describe in a minute. And that was developed under ACE Nano, which was a project that ran for 4 years and just finished last year. And there we developed a workflow so that the people doing the experiments could document how do they prepare the sample, how do they carry out the measurement, what's their data treatment. And then the idea is all data that is generated with characterization work should have an associated protocol, even an emerging new version protocol for a new method that eventually becomes an SOP. So this idea of documenting linked protocols to linked data, I think, is quite an important one. And we're also working on that, for example, within Risk Hunter. How do we document in silico protocols? So there, we're currently working on how to do that for QSARs, toxicokinetics, and use of omics data and points of departure. Again, I think a good area of work that fits, more open collaboration around that work. And this has led to this NanoCommons Knowledge Hub, which tries to bring these different tools, data and workflows together, and also provide, importantly, increasing amount of training activity and guidance so that it's easier to figure out how to actually use the tools and use the data. And then I just finish up on a current initiative, and we started this in practice last year, where we went through a process to register as a virtual organization a couple of these activities like NanoCommons and OpenRiskNet under an EGI project, which is aiming to help put in place a structure and processes for enabling community members like us, collaborations, projects, developing tools to actually deploy them in a sustainable way as part of the European Open Science Cloud, which is what this EOSC is, European Open Science Cloud. You'll probably see that acronym quite a bit in coming times. And our goal here is to establish a sustainable, collaborative, open knowledge infrastructure supporting predictive toxicology and risk assessment. So in the initial work, we've set up a computing cluster, some processes and architecture for orchestration. So we use things like Kubernetes, and we support, say, partners or organizations that have developed a software or a database. It needs to be containerized and then deployed on the cluster. So we've made a start on that with about 10 resources from NanoCommons as part of a transnational activity in the spring, and now we're working on how to extend that to other areas like the EU Tox Risk knowledge base, OpenRiskNet services, ONTOX resources, and so forth, and to support that, to have people supporting that kind of deployment work and helping partners deploy their software. And we want to make that collaborative. So if you have resources that you would like to be part of this, or you have someone on your team who's technically able to contribute and be involved, we want to make it as open and as collaborative as possible. So currently, there's some initial deployment workflows in place. There's certainly plenty of room for further work, but the idea is we could put a standardized configuration, testing, deployment workflow in place, including updates to software, and make that sort of robust, an industry kind of standard kind of workflow for deploying software and databases in a reliable way. And I've just recently set up a survey, reaching out to different organizations and people in the community I know are developing interesting resources and supporting this this kind of open science work. And I've set up this form, which I'll share with you in the chat, because anyone interested at this early stage in getting involved in some way, you can express your interest on this form. It's not a complicated form. And I'll share that in the chat after I finish, which is now. And there's my contact information. You'll find that I post updates at times on LinkedIn, so you can follow that, or you can also fill out this form if you're interested in being part of this new work. And that's it for me for this morning. Back to you, Andreas.
Excellent, great. Thanks a lot, Barry. Please put that into the chat, so because then we can make that interactive a little bit this meeting as well. Feel free to do that. Are there any questions or any comments for Barry? I would have one regarding the components you mentioned from ONTOX in the beginning. How are they actually accessible to others? Is that some type of subscription model, or is Edelweiss then a partner in Consortia and this is how they get used? Or I mean, basically, how does that get disseminated, in which way?
Yeah, you know, it's kind of being sustained, I would say, more conceptually and with further iterations on in different ways on different projects, including the ones I showed. So it was never really just the way it worked out in terms of the history. OpenTox didn't become sort of a sustained resource on keeping those APIs updated in a centralized place and so forth. So it's been kept, I think, alive more by different kinds of applications, by different people and different projects. I mean, it does raise a question now that we're, I mean, a decade later. I mean, it's definitely a different world. We've got a lot more tools, a lot more data, and we have this, I think, good timing and potential now for this deployed knowledge infrastructure. I mean, that would raise the question, could we revisit that goal of having, say, some kind of community-based mechanism of sustaining useful things with regards to APIs and metadata and so forth, and that potentially could be part of a funded initiative. I mean, related to this initiative, we'll certainly look to apply for funding. And I think that could be, with regards to infrastructure funding, to support a sustained community-based resource on common kind of knowledge objects like APIs and ontology metadata templates could be very valuable, because currently there isn't really, it isn't really well organized enough and well funded enough. But I think that would be very worthwhile.
Yeah, I agree. That's exactly why I asked. So sustainability is often a problem there, right? So if there's anyone listening here, get in touch with Barry if you have any input or interest in forming Consortia. David.
Yeah, thanks. Hello, Barry. Thanks for this nice presentation. I would be curious to know a little bit about your views about this emerging standard on about data sharing in Tox called Euclid. And what I don't want to right away provide my perspective, but I would be really interested to get yours.
Yeah, so now we get into really the topic of the the regulatory acceptance of the new methods, because we need to pay attention to the guidance and the templates from regulatory agencies, OECD and EFSA and ECHA and so forth. And it's important. I mean, we have to do that. So when I'm looking at now our approach on documenting in silico methods within Risk Hunter, and it can be broader than Risk Hunter, I do pay attention to that. So I'm paying attention to the emerging OECD guidance on transcriptomics reporting. In QSAR, it's fairly straightforward because it's established. We have a QMRF modeling reporting format. We have a QSAR prediction reporting format. There's guidance from the European Chemicals Agency. So there it's quite clear because, I mean, people have been building QSARs for a long time. But when you get into something like toxicokinetics, it's much more challenging because there's still emerging approaches to be developed. And now your Euclid question, we of course have to grapple with that because Euclid's a very important reporting format. I think the difficulty we face is how do we have good practices that work in research without being so heavy that they really overwhelm the research group in terms of the kind of detail a regulatory situation is looking at. So somehow we need to align it, but we need lighter approaches to research. I mean, people need to get research done and publish their papers and move on to the next problem, and they don't necessarily have weeks of time to fill out such detail. And they complain about even documenting protocols because they say it takes too much time. Can you make it faster? So I think the trick may be if we can try to figure out how to align in a sensible way the research outputs with these regulatory formats, and then provide tools so that we can better document the data and the protocols. And then if we can align it with metadata, let's say Euclid or the new OECD guidance has, we get closer. But I don't think we want researchers to always have to use very heavy reporting tools and formats. So it's going to be figuring out the balance, I think, on that.
Okay, thank you.
That makes sense.
Makes totally sense. It's super complex topic and common language. It's all about being able to interact and make the data accessible to everyone and usable. I mean, making them in a fair format. But of course, there are some huge complications and it's super complex. Yeah. But thanks for this.
There's a question from Amsala. What do you mean by animal-free safety assessment of chemicals? I think anyone could answer that, but basing my understanding of that with regards to a program like say ASPIS is we would not generate any new animal data for the purpose of the safety assessment. We would try to use in silico data, models, in vitro data, and of course make the best use of existing animal or human data if it's available. So the tiered strategy wouldn't involve new animal testing for the purpose of the assessment. Do you agree?
Yeah, that's right. I mean, sometimes at conferences you have people who are new to the field, and that's why you get questions that. But it's good to, I think, underline the ideas of the field again. No, that is right. It's a big trend right now. I would like to add, right? It started with consumer goods, right? So where you were not allowed to do animal testing, I think for 10 years now. Agrochem, right? In other areas, there are mandates to phase that out in 2030 or so, right? So it's a big development over decades, actually. Yeah, it's a very big direction right now.
So my final note is I'll just post this form in the chat if you're interested in being involved in any way. The survey is mainly aimed more at people developing resources, but if you're also interested as an end user as well, it's also fine. But it's mainly just trying to see who's interested in being involved in this, and then we'll follow up and try to organize some communications and discussion, and hopefully some people want to be involved in deploying a good software or a good database that they have.
Okay, excellent, great. Thanks a lot, Barry, for your contribution as well. Much appreciated. And for you as well as the other speakers, feel free to send me the slides as well. I just put Mathieu's slides on the website as well. Feel free to send them over because they might be useful for the audience. Yeah. Great.
And do you want the speakers to send that to you by email?
Yeah, just send it by email. I will convert it into a PDF probably and put it on the website then, actually along the presentations. Yeah.
You'd prefer a PDF then?
PDF is good. Yeah, PDF will be great.
All right, we'll do that.
Excellent. Great. Thanks a lot, Barry. Cheers. Okay, we'll move on to our next speaker, and that's exactly the consortium that was mentioned by Mathieu already, and that is the PrecisionTox consortium, which Xiaojing will guide us through. Feel free to share your screen. Very welcome.
Yeah. Hello, everyone. I'm Xiaojing Li. I will now share my screen for the slides. I will be the one that introducing you about our precision toxicology project. The precision toxicology that we talk about today will be covering their basic ideas of the whole projects, also focusing on how we would like to discovering the molecular biomarker of chemical hazards. I will share with you one of the case study that I have done with the same methods and also some conclusions over that case. The precision toxicology is a 5-year project funded by the European Horizon 2020 research program. Our teams in the University of Birmingham are teamed up with other 15 teams across 8 countries trying to pursuing these grand ideas. The main goal of the PrecisionTox project is to develop a new methodology which will be help us better protecting the health of human and also the environment. It will establish a new set of names, which will be our new approach methodologies for the chemical safety testings. These names will aim at revealing the causal relationship between the chemicals and their adverse outcomes. There are mainly 3 pillars in our projects, which are phylotoxicology, variation in susceptibility, and also embedded translations. For phylotoxicology, this idea is to focusing on replacing traditional animal testing by an array of bioassays. As no single testing species, including mammalid species, is the perfect surrogate for the humans or other species, we propose to use evolutionary diverse model species plus human cell line to identify the fundamental biological mechanisms under perturbation like chemical exposure. Once we have some ideas of how diverse and conserve these chemical toxicity response across multiple species, we'll move on to determine the variations within populations. Ideally, with the information of chemical mode of actions discovered in the phylotoxicology and the linkage between genetic targets and individual susceptibility, a more precise estimations of the same exposure levels can be determined. This project is not just for delivering scientific findings, it is also about regulations. We are interacting with stakeholders really intensively to try to know about their concerns of the chemicals to be tested and also how we want the new methods to cover. The case study will integrate our scientific findings into regulation practice. The overall joint efforts for these 5 years will cover a total of 250 chemicals. Those chemicals, some of them might already have AOP information, some of them might be the new emerging chemicals in the field. So we try to covering as much as concerns that we could have. The general results for these whole projects will be a new regulatory paradigm of detecting toxicity. with the NAMs focusing on generating comprehensive profiling of the biological responses, we can produce data to construct mechanistic understanding of how chemical cause harm by avoiding animal costly testing methods. Today, I will mostly focusing on the final toxicology part. As said before, a traditional testing species may not be the perfect surrogate model for human toxicology or even easily extrapolated to other species that are not tested before. A whole organism toxicity testing is preferred in the toxicity studies and definitely crucial for better understanding of toxicology. We propose to use an array of bioassays with non-traditional test species. They are evolutionary diverse. For example, Daphnia magna, which is a crustaceous, a keystone species in the aquatic system, Drosophila and C. elegans being biomedical surrogate models. The zebrafish and the xenopus would be our perfect model to capturing the developmental and hormone-related responses, and also human cell line as the surrogate model and in vivo model for the human itself. Some of these testing systems are already recommended for chemical testings, for example, like Daphnia and zebrafish. Our teams in the Birmingham is focusing on multi-omics profiling of the Daphnia magna. These keystone species have been well studied for many years, but we are now trying to understand a bit more with the emerging genomic resources and also a lot more detailed databases and their metabolomic profiles. We are trying to developing new methods for the phenotypic profiling as well, so we can actually understanding all these biological features at the molecular level and also individual levels. Our efforts in developing MEMS will also include computational methods for integrating multi-omics data and data-driven models to identify those molecular biomarkers. You may be familiar with Aop Framework, so I will briefly touch on this. The adverse outcome pathway has been revolutionized the field of toxicology. This framework aims at linking evidence generating from different biological levels to serve as a knowledge assembly. And AOP may represent as a series key events, starting from a molecular initiative event and end at an adverse outcomes. These key events may be represented by biological features depicted at multiple levels. With the advancements of high throughput sequencing and high resolution mass spectrometry, we'll be able to generate molecular profilings for transcriptome and metabolomes, which enable comprehensive profiling of biological responses at the molecular level. These provide an opportunity to identify molecular key events more effectively. Within PrecisionTox, the molecular key event is defined as a set of interacting genes and metabolites, which may represent as a sparsely connected networks. These biomolecular features are necessary in relation to an adverse outcome. So with those two efforts, with evolutionary diverse model species testing array to allow us constructing data for final toxicology, and with the multi-omics profiling that allow us to identify molecular key events induced by the chemicals in all these model species, we will be able to summarizing the elements for the molecular key events that we want to identify within our projects. We will be summarizing the molecular key event as three characteristics. The first one is the molecular key events are induced by chemicals, which means they are highly closely related to the chemical exposure and the exposure level, the time, and also the concentration levels. They may link to a hazard, which is either observed from the phenotypic traits or observed from the biological interpretations over the molecular features. They can also be shared by multiple species, which are interpreted by their evolutionary conservativeness. These three characteristics allowing the fundamental understanding of the chemical mode of actions, which are shared by a suite of species. so may as well be our biomarkers of the chemical hazards. If the molecular key events is shared by non-traditional testing species like Daphnia and humans, the mechanistic understanding described by the Daphnia may work as well for the humans, which build a link between our human toxicology and eco-toxicology. In the end, results generated by all these 5 distantly related species may advise on the potency of a shared chemical hazard. Risk managers and chemical regulators may receive this knowledge on the likelihood that human can be harmed by certain class of chemicals only because the humans have processed the same toxicity pathways as other distantly related species by evolutionary descent. Now, I will briefly talk about our strategy over how to discover the molecular biomarker of chemical hazards. It will include a total of 4 steps. The first step will be generating biological profiles, which in this case would be our multi-omics profiles. Our teams are focusing on transcriptome and metabolome. Transcriptome would be describing how these genetic features being expressed and the levels of them. And for the metabolomes, what has happened, which would be how much metabolites being accumulated with the system. To construct a biomolecular networks, we may need to use certain computational methods. One of the computational methods have been developed long times ago would be the co-expression networks for genes. We're now trying to using the same methods for both the genetic model, the transcripts, and also the metabolites. These co-response modules are constructed by correlation analysis in between genes, which will include Pearson, Spearman's, or mutual informations, and also module identification algorithm to help divide those networks into modules. Genes or metabolites within each of the modules are correlated in certain ways. Either they are functionally related, they are co-responsive, or even they are co-regulated by a certain regulation factors. After regenerating the informations for the network, we are now trying to performing an association analysis between these 3 elements. The first elements will be our chemical stressors, which will be performed by a single factors for their chemical concentrations or even time of their exposure. The second element would be our modalities for the omics, which could include certain matrices. These matrices could consist of a transcriptome matrices or multiple metabolome matrices. And for the phenotypic traits, it could be quantitative features to represent the hazard levels. Ideally, we could include all these matrices into one single model, and by using multi-block correlation analysis, such as sparse canonical correlation analysis, we could generating the hypothesis by using a data-driven model to extract the covariant features within those modules. The 4th steps would be to identify the molecular biomarkers. Ideally, after the correlation analysis, we could extract which specific set of biological features actually co-varying along with the chemical stresses and the chemical hazard. By using ortholog-based annotation, we could identify which specific features are actually conserved across multiple species. By summarizing those information at the pathway levels, we could even try to qualitatively assess how those features are conserved across multiple pathways across the species. This information will allow us to understanding whether those responsive features recorded in single species are actually functionally conserved across multiple species. The results coming from these models could also be used for chemical classifications or even performing probabilistic modeling for the hazard potency. Our overall goal is to try to develop the toolkits to identify the molecular biomarkers for chemical hazards. Now I will show you one case study that I have performed before, which is using the real river samples and environmental samples. And we'll try to identify which corresponsive biological features are associated with the chemicals within those mixtures. Those water samples are collected from the Danube River. There are a total of 12 sites. All those sites are contributing to the water analysis, chemical analysis, which including polar organic substance within those waters, and also serving as the exposure media for the exposure test. The chemical targeted screening analysis, including 457 chemicals that are references. We could use those references to help us performing semi-quantifications over those substances. In total, 91 chemicals are detectable within those 12 sites. You can see from the right-hand side plot, the PCA plot showing that within those 91 chemicals, the general distributions of those 12 sites are looking like this. One of the things that is quite obvious is the D11 site is departed from the other sites. So we try to understand why this happens. And when we calculating the contributions of each of the chemical substances, we find out the carbamazepine and their transformed products might be the cause. These 4 chemicals are detected in all 11 sites and at the relatively high concentrations, especially in D11 sites. These 4 chemicals are actually under detection levels. So we think these might contributing to these distribution patterns. The chemical and their transformed products are these 4, which are colored in blue. So these 4 chemicals we actually detected within our organic mixtures in the waters. While these figures is actually adapted from the KGG map, which is characterizing how carbamazepine are transformed and metabolized within the human systems. So we actually know that within those waters, it includes both the parent chemicals and also its child products. We are performing the OECD 202 acute immobilization test with our Daphnia magna. These Daphnia magnas are neonates within 24 hours. They were exposed to those waters for 48 hours, and then the immobility will be counted. All those exposed tissues would be collected and flash frozen for multiomic extractions, which means one single samples will be both used for the transcriptome and metabolome. The metabolome are including 2 assays for the polar metabolites under positive and negative ion mode. For our second step in the workflow, we will generating the co-expression networks for each of these modalities. For example, for the transcriptome, we have in total of 19,000 features. And after constructing the co-expression networks, we have a total of 11 modules, which are listed in the right-hand side. The maximum modules are including 1,200 genes, while the minimum one are including 20 genes. Each of the module will consist of more than 5 to 10 different pathways, depending on their pathway enrichment analysis. Association analysis between all those 4 blocks are including those chemicals data for those 4 chemicals, the RNA transcriptome data, and also 2 modality for the polar positive and polar negative for the metabolomes. By using sparse CCA modeling, we could actually establish a varying models for those 4 modalities. You can see from the right-hand side, the components for the transcriptome are accounting for 6.4% of the total variations in the transcriptome data, while the other 2 modalities are accounting for 7.2 and 9.4 of the total variations in polar negative and polar positive, respectively. The general distribution could also see that the D11 is far behind, which suggested that the covariations are actually captured by these sparse CCA models. With the pathway enrichment analysis, we can identify which pathways are involved in those modules. For example, for the RNA module M1, it consists of these 7 pathways, and one of the pathways which I have highlighted in yellows are the drug metabolism pathways, which are accounted for the carbamazepine metabolism pathways recorded in the KEGG database. We could also analyzing those pathways by their KO terms. The KO terms would be their functional orthologs recorded in the KEGG. We recorded how many KOs within each of the pathways for Daphnia, which is listed in the first column. And we also try to compare the compositions of the KOs with other species in our projects, which are the C. elegans, Drosophila, Daniel, Xenopus, and also our humans models. You can see from this digits that there are high levels of conservedness within those species. By saying that, I could only say these data are qualitative assumptions based on the Daphnia magna. Ideally, if those 7 KOs are the key functional and canonical functional units within those pathways, we will assume or suspect that the other species might respond similarly with those as the pathways are conserved because of those 7 KOs. But we definitely need experimental data to help us understand whether the same chemicals will induce the same set of KOs within those pathways. So for this case, we could actually forming the information looking like this. We have a chemical set in request that are associated with 10 sets of corresponds modules. These modules are significantly enriched in these 12 pathways. These pathways ideally are including the ones that reflected of their bioactive activity, their biotransformations, and also certain hazards related to those chemicals. So for this case study, we could actually identify the molecular biomarkers that are associated with carbamazepine and their transformed products. We find out the true positive, which is the drug metabolism pathways, but also some others that might suggest that chemical hazards. These suggested that the data-driven modeling strategy can also actually pull out the informative molecular biomarkers. The pathway conservation analysis shows that the KOs in the response pathways in Daphnia are highly conserved across multiple species, suggesting similar exposure response may be expected in other species, including humans. In our PrecisionTox project, we will provide experimental evidence to support these assumptions, whether these responsive biomarkers found in different model species are actually functionally conserved. In the end, we want to use an array of model species to try to construct the mechanistic understanding of the chemical mode of actions. For example, for Daphnia magna only, we could using the information directly to interpret the ecosystem toxicity hazard, but we could also use the pathway conservation to suggest whether they will form harms to human as well. This is whole ideas would be captured by the toxicity by descent ideas, which being just published like a week ago. So that would be all the topics that I would like to cover today. I would like to thank my teammates from the University of Birmingham, all the colleagues and teammates from our PrecisionTox project. I would like to thank the organizer to invite me for this talk and also the audience for your attention. Thank you.
Excellent, very interesting, Xiaojing. Are there any questions from the audience? One comment from my side, I quite like the phylotox approach that you describe. There are many advantages to that. Yeah? One is I've realized in the past hypothesis-free data generation. Yeah, if you just generate large-scale omics data, yeah, it's very, very painful to find a signal, a meaningful signal in that. Yeah, that's one reason. The second one is, of course, if you have understanding, you know which species you can use as a model species. Yeah, which data do you need to generate? You can extrapolate. Yeah, so you need both understanding of the system and data to fill that. Yeah, and I think, and I usually consider it this way as well. Yeah, you need really both. Yeah, not hypothesis-free data, but some understanding, and then you need to fill it with data, with the right data for the right purpose. Yeah, so I quite like that PhytoTox approach that you described in the middle of your talk.
Yeah. So for the PhytoTox, we are trying to actually integrating both the database that are already existed and also experimental evidence. For the database, I could cover a few observations that are just being performed. For example, for the Daphnia magna, we have a total of around 30,000 genes. However, only around 7,000 of them are actually have certain functional annotations, which is like really low. And some of the other, even for like humans, the maximum annotation rate would be only like 50 to 60%. And if we consider in pathways, it would be much lower, like 10 to 20%, which means if you're only using one single species, you can't understand a lot more about their functionals. So we definitely need to integrate both the knowledge that have been existed in the database and also some other evidence that are coming from our new experiments.
Yeah, there are many good reasons to do that. And that's right. Nadav, you have your hand up. You have a question?
Yes, thank you. Yes, first of all, this is a really interesting work, and I like what you did there. I would ask, please, how would you account for a scale? I mean, you would like to maybe repeat this process for countless chemicals, which will present very difficult challenges. So I was just wondering, how would you face that challenge?
For the chemicals part, at least for the first stage in our, like the first 2 years, we're using chemicals that already have the AOP information or even have well-studied mechanistic. So the way to do that is actually to find whether the knowledge that we already have could actually be discovered by our data-driven models, which is try to proving that the data-driven model is working, and also to try to summarizing whether these knowledge coming from one single species, for example, the original data are actually generated by mouse or even humans, but somehow they could be actually reflected by these distantly related species as well. So we can actually have a more systematic understanding of the final toxicology, which help us improving the toxicity testing by understanding their mechanism at the molecular levels.
Thank you very much.
Thank you.
Okay, is there any final question from the audience? Okay, then great. Thanks a lot again, and all the best for your project, Xiaojing. Thanks for your presentation. Yeah?
Thank you so much.
Thank you. Okay, great. And our final speaker in this session will be David Rouquie from Biocrop Science. And so he will exactly present one concrete consortium, how novel types of readouts can actually be used for safety and toxicity prediction in the future. David.
Hello, everyone. Thanks, Andreas, for the kind invite, and and also to animate this community of people interested into the in silico toxicology. I think we have a lot of work to be done ahead of us. And I think it's super interesting to exchange and to share ideas. And thanks again, Andreas, for organizing these discussions. So today I will fly quite high in terms of altitude into so I won't go into too much of the details. I will be very, very much conceptual. And it's about describing an emerging collaborative project, which is elaborated into an organization called HESI. I give a little bit of information about this organization later on. And it's on the use of molecular tools. So in that case, I will be referring to cell painting data and high throughput transcriptomics in the context of next generation risk assessment. And I'm gonna describe that a little bit later. So the agenda is, so I'm going to briefly introduce what's going on in terms of regulatory environment when it comes to chemical safety, chemical risk assessment. Then I'll describe a little bit the research proposal, few words on HESI, and actually it's a call for contribution. So we are eager to get people involved into this collaborative efforts. And so please reach out. I'll provide the contacts afterwards. So looking at what's the driving force or the regulatory context. So there have been a strong call starting from US EPA, US EPA is the equivalent of FDA in the US for the plant protection products and industrial chemicals. And there was a paper back in 2019 stating that we should go into this direction of elimination of all animal testing, and quite soon, because it's by 2035. And so you can imagine that it's something for the chemical industry, which is something serious, and that we need to take care and to adapt to this situation. So Europe, of course, these things is also coordinated, and Europe even provides some recommendation.
David, just to check, are you meant to share some slides? Or?
Yes.
Oh no, you didn't yet. Please do.
Okay.
Yeah, now we can see your screen now.
Okay. Yeah, I think it will be a little bit easier like this.
Yeah, very good.
It's okay now?
Yep, now it's good.
Okay. So yeah, so US EPA stated in 2019 that by 2035 we're gonna kind of ban the animal testing for risk assessment of chemicals, and in Europe we have this strong call for also to already engage some modification in the way we are performing the risk assessment of the chemical even earlier, because there are some roadmap mentioning some actions by 2027. So the driving force for this is not only the obvious ethical reasons about just stopping the animal testing, but also it's targeting the objective of improving the risk assessment of the chemicals, in the sense that instead of going through information generated in lab animals and then going into the human situation, it does imply a cross-species extrapolation where you get some uncertainty, and so the idea is to go straight to the human situation in order to do this risk assessment. So you see, very big question, very conceptual questions, and one can see that as a long and bumpy road ahead of us, and in a rather short period of time. So just to give you an idea, when you're in R&D in industry, typically a molecule you developed it in at least 10 to 14 years. So it means that the hit or the molecule we're at the moment discovering in our pipeline will be impacted by this decision on the regulatory context. So to this long and bumpy road, we need to make sure that we can learn how to ride it. And so before being in this situation where we are mastering this the ride, we probably need to go step by step. And for me personally, I think we are still at this very initial step with this child bicycle within small wheels, where we need to make some learnings. So before getting to the point that we have a fully validated suite of new alternative methodologies that are going to be applied in the regulatory decisions for based on this new way of doing risk assessment, we still need to make some conceptual learnings, in particular when it comes to the in vitro to in vivo translation. And this all about these conceptual learnings is exactly what we are targeting in this proposal. So looking a little bit into the data landscape and what is available at the moment what has to be generated in terms of data for achieving this objective in terms of toxicity prediction. So I'm going to present this data landscape making use of this pyramid, assuming that you have in x-axis the amount of information required. So the more you go up, the less you get information. So we had, starting from the bottom, a huge amount of information when it comes to compound chemical structures in the range of trillions even. Then moving up, we get some information in terms of about those molecules, about on and off target assessment. So about mutagenicity, for example, or some bioactivity information related to toxicity. So in our case, in agrochemicals, it's steroidogenesis or some endocrine effects. Then moving a little bit up, so we are falling into this domain of identity biological responses. So high throughput transcriptomics or cell painting are part of it. And on the tip of this triangle, we have in vivo outcomes, so have access to the in vivo data. And in that case, we are lucky at Bayer because we are still the two division between the crop science part and the pharma part. So we can share data, in vivo tox data. And when it comes to the preclinical, so the lab animal data, and there are some clinical data which are even more even less accessible. But this is the key elements certainly to get access to those data to anchor these new methodologies. And so of course also there are some public data sets. I cite one of them from the US called ToxRefDB. So when you look at what has been done so far and what we know that we can make some different types of jumps. We can move from the compound structure and make some prediction using machine learning or rule-based methods, in terms of on and off target assessments and gain this knowledge about the properties of those molecules. We also know that we can move down from the high-density biological responses, could be transcriptomics or cell painting, to infer these properties, so this target or molecular initiating event, as we like to call that in toxicology. And the objective of the proposal will be to make this jump upward, to make use of these high-density biological responses in order to assess the predictivity of the in vivo outcomes. So I have to talk about this elephant in the room, as I like to call it. It's about everything dealing with the ADME properties of the molecule. So everything about the kinetic properties of the molecule, which of course in vivo it plays a super important role, but it will even take a super important role when it comes to new alternative methodologies as well. So the objective of the proposal. So the objective of the proposal, maybe a few words about the history and how come this idea emerged. One of the driving force was some of the comments I heard from this, I think it was Mathieu or Vrije, I cannot remember, who mentioned this scientific and regulatory advisory board we have in this European project. So we are part of the Risk Hunter project. And one of the advisors was saying, yeah, whatever you want to do on the NAMs, for us, the key aspects is about the false negatives. So just make sure that if you have a battery of tests of new alternative methods, you need to show us that actually you don't miss a bad guy, which going to trigger some public health effects in the future. So we need to benchmark this and to make sure that the development of this technology, these new technologies are then robust enough do this new risk assessment. And so the idea was to say, yeah, in order to evaluate this rate of false negative, then let's make use a large collection of molecules and then assess that. And in order to get these big numbers, then the collaboration and data sharing was a key component. And so the intention there in this proposal is to make use of already existing in vivo data that has been generated in industry, so in AgChem sector and in pharma. So we are talking there about non-genotoxic compounds for which we have already the information about what the molecule is inducing in lab animal, and in that case it will be in the rat to make it very very specific and also realistic in terms of development. So we want to make use of those molecules to benchmark the data generated in vitro using cell painting and high-throughput transcriptomics in order to, again, establish and validate this technique and see how robust they are, how useful they can be in terms of NAMs. And the idea is to make use of the hepatotoxicity domain as a type of toxicity as a use case. So the overall project tagline is the following. So we're going to start from the in vivo effects observed in animals and using all those compounds for which we know what they are doing in the liver of the rat, and in that case it will be in a subacute study. We're going to classify the different histopathological findings and by the way the chemicals into the different types of effects one can observe in a liver in a rat following chemical exposure. So we know already that depending on the chemical space and even the indication could be agrochemicals or pharmaceuticals, we don't have exactly the same type of effects which are induced by these different types of chemicals. So that's going to be also the way to explore that into more details. So having this in vivo study, so we have this qualitative information about what are the effects in the liver, and we have also some quantitative effects information when it comes to the dose level at which those effects has been starting to be observed, so the no effect levels that are going to be part of this. Of course, we are also looking about ADME properties of these molecules, and that will be super important. And from there, we're going to profile, starting from the rat cell cultures, the molecules, and making the full transcriptomic profile of those ones in a dose response manner to determine the point of departure, then have an estimate of the exposure of those molecules in an in vivo situation, and then be able to compare the two situations between what the in vitro test system is telling us compared to what we know what's happening into the rat. The same will be done using cell painting, and the same will be done using human cell lines or cell culture. Okay, so both using different types of the two types of technology, transcriptomics and cell painting. All this to combine this and try to do some comparison, conceptual comparison, I would say, out of the data generated. So what could be the types of comparison we have in mind? So we have some quantitative aspects we want to compare. So that will be the comparison between the in vitro to in vivo situation in the rat. That could be also the in vitro to in vitro, but from rat to human, and see what does that tell us. And then there is this what I call qualitative or mechanistic aspects. How consistent the effects mechanistically are related between an in vitro rat cell and an in vivo rat cells in a given animal. Then there will be different types of comparison between in vitro rat and in vitro human cells. And if we are lucky enough, we will be even able to get this comparison between in vitro human cells and in vivo human findings if we manage to get this information from clinical data. Those are the two big aspects, but we think that it's going to be also a nice way to make use of those data for MIE prediction, read across, and in the future maybe for AI-guided de novo design of the molecules. So a few words rapidly, because I don't want to, yeah, I think it's still okay, but a few words about in which frame we are doing this exercise. So we are doing it in the frame of HESI, because the big advantage of HESI is an organization. It's a scientific organization gathering all the stakeholders involved in all those discussions. So we are dealing there with academia, government, industry, and scientific and other scientific organizations. So, and it's a super impressive organization, very productive, and which has done a lot of work in the frame of chemical toxicity in general and safety safety aspects. So more precisely, there are different subcommittees within HESI, and the project is evolving in the emerging system toxicology for risk assessment of risk, (ESTAR). So it's a subcommittee, and it's purely on the focus of this subcommittee, because it's about development and delivering innovative system toxicology approaches for risk assessment. And so just a few words about where we stand in terms of a working group. So the working group is established. We have regular discussions involving different stakeholders, companies, pharma, CROs, academia, and government agencies. And we are at the stage where we are discussing the overall study design and looking for options for funding, and also make use of in-kind contribution at some points. And we're going to have a dedicated Alpha Day session, early November, to discuss into more details the detail of this proposal. This is the call for contribution. So if you're interested, please don't hesitate to reach out to Sadef, Connie, or myself. And as an outreach and a little bit as some sort of advisement, we're going to have a session at the SOT with the great honor to get Anne Carpenter delivering a talk, and also Josh Aril, and also Jessica Larocca, and Deidre Dalmas, and Fabrice from IT. And with this, I'd like to thank you for your attention. And again, if you are interested, please reach out, and you need an active contribution of everyone. This is really a collaborative effort. Happy to get in touch with you.
Excellent. Great, David. Much appreciated. And John already started with a question. John, do you have a mic, or should I read that?
Yep, I have a microphone. So my question was whether which cell lines did you use in your study, and whether you spent much time kind of thinking about which ones and maybe optimizing or testing on alternatives.
Yeah, so that's a great, that's a super important aspect indeed of this proposal. Nothing is set at the moment because it's it's in the making. There are conceptual questions there for me as well about the physiological relevance of different cell lines compared to a specific adverse outcome. There would be also some conceptual comparison to be made between cell line and primary cell cultures. So we're going to try to tackle all those aspects and make all the learnings we can make. When it comes to cell painting historically has been developed by using a cell line called U2S. It's derived from an osteosarcoma. So it has nothing really to do with a liver cell. But it's part of the benchmark is to see how much the biological processes which are captured by this cell line because it's an easy to go cell line to do the cell painting. Can inform about different types of toxicity and different types of pathways which can be present or expressed into the test system. So yeah, we know cell painting in the case of U2S, there is for example, the estrogenic pathway is not expressed. So if you are looking after a molecule which will trigger or induce on stuff in this pathway, then we can just simply forget about it. So, but that might be something. Yeah, that would be all the learnings we're going to try to make between, and the physiological relevance of the test system in vitro is essential. Yeah.
Yeah, I think that would be really fascinating to see the results from that. So, thanks very much.
Yeah, cheers, Don. Any other questions or comments from anyone? Yeah, in my experience, so we worked on gene expression data as well as cell painting data before as well. I think what we didn't evaluate yet, and that's exactly where this consortium, for example, fits in. Yeah, it's kind of the applicability domain, right? So what do I actually see in the data I have, right? And that means three things. So one is the chemistry, then you have the mode of action, then you have your in vivo relevant endpoint, right? The one you're actually interested in. Yeah. And quite often we don't know that. And sometimes there's validation on levels. Well, you have three levels, right? One is something that's obvious, right? Tubulin inhibition, let's say, right? I mean, you see that in microscopy, right? But it's not particularly interesting. It's very high level, it's an extreme effect, if you wish, right? But the other parts are where there's something more fine-grained, right? Do I get that level of resolution as well? And where don't I actually see anything, right? And to discriminate which type of activity actually falls into which bucket, yeah? So where do I have actionable information in the data, yeah? I think quite often we didn't evaluate that yet. And sometimes if you have publications, yeah, there's a focus on the more obvious cases, yeah, but not so much on the final level of detail that we quite often need for practical decision making, yeah. But this consortium is exactly one step into that direction, right? So what do we actually see in the data we generate, right?
Yeah. No, sure. It's again, this physiological relevance is essential because yeah. If I oversimplify some of I mean, these initial ideas about this next generation risk assessment, there have been this idea that yeah, that's easy to go into the NAMs because so you ban animal testing, then you go straight into a human cell line, and then you get your information about the human cell line. So we've done some comparative analysis about the outcome of the signatures and the transcriptomic level between and I think Benoit is in the call. So Benoit Baif, he was working in my team in the past. He did show that even if you take a cell line which is derived from the same tissue, so in that case it was lung tumors or because it was even tumors. Or prostate tumors, you don't explore you really don't explore the same biological space in this situation. And then comes this tricky question about the chemical space, which is also coming with its own constraints.
Yeah, no, absolutely, yeah. Yeah. Any other comments from anyone? Any questions? Okay, great. No, then thank you to all the speakers of this session, this consortia session, and it's meant to be interactive. So if there's anything to follow up with any of the speakers, please do get in touch with them in order to bring the whole field forward. We will have a brief break until 10:30 UK time, and then we will start with a session on modeling approaches and so on. Okay, see you in 10 minutes.
Thank you.